Hello and welcome everyone. In this podcast, I'm going to talk to you about some of the basics of epigenetics. That will set the stage for the next set of podcasts where we're going to talk about transcription, and then for the rest of the semester where we're going to talk about the molecular events of epigenetics and then how it is ultimately linked to gene expression and traits and diseases. It may take more than one podcast, but what we're going to talk about is first, how do we use our genome? And then I want to move into a brief history of epigenetics. And then introduce what we mean when we say epigenetic marks. And then I want to talk a little bit about the types of messenger RNAs. Not in great detail, because we will come back to some of these throughout the semester, but it's a good idea to set the stage of the kinds of mRNAs that we might move into this semester. And then finally we will talk about the importance of epigenetics. Just real brief, because we could spend an entire semester talking about the importance of epigenetics, but just to kind of set the stage. Okay, so now let's focus in on this big question of how do we use our genome? We carry it around with us all the time, but how is it that we use it? So first let's talk about HIV. HIV has less than 2,000 base pairs. Pretty puny as far as genomes are concerned. Next, let's talk about E. coli. E. coli has 4.6 million base pairs. So pretty big jump there. Then let's bring in our first eukaryote, yeast, Saccharomyces cerevisiae. It has 12 million base pairs. So let's talk about C. elegans, another common model organism. This worm has 103 million base pairs. Now let's look at Drosophila. Drosophila, or the fruit fly, has 170 million base pairs. Here's a picture of a trumpet lily, and a trumpet lily has 90 billion base pairs. A lot of base pairs. So let's bring in our last two, humans and amoeba. So amoebas would appear to be a fairly primitive eukaryote, and humans were a fairly derived eukaryote. So we might think that these two could set some extremes of genome sizes within eukaryotes, but what we find are that humans, we have almost an embarrassingly small amount of DNA, 3.2 billion base pairs. I mean, compared to yeast or C. elegans or fruit flies, it's quite a bit more, but compared to a trumpet lily, we have far, far less. And shockingly, when we look at our friend here, the amoeba, it has, hope you're sitting down, 670 billion base pairs. So a couple take-homes from this is that we can't necessarily predict the size of a genome based upon what we think about the complexity of that species. The end result is genome size really means nothing when we're thinking about an organism. It's just a number. But a common trend for all of these organisms from HIV to E. coli to trumpet lilies, amoebas, and humans, is that they all have to be able to package their genome in a certain way so it can be used. And so it has to be packaged efficiently. So let's move on and talk about how the genome is used. It's important to realize that the genome is not just randomly shoved into the nucleus, or in E. coli's sake, randomly shoved into the cytoplasm. Rather, it's placed into the nucleus in a very usable manner. And I think a nice comparison is if we were to look at a very unorganized collection of books. This is a picture, hopefully it's big enough that you can see, but what we have here are a couple bookshelves. Some of the books are on there, and then we have a whole bunch of books just scattered throughout on the floor. So this would be, in our comparison, an unusable collection of books, which we might think of as, in our analogy, unusable genome. If I had to go into this library and pick out a book, for instance, if I had to go to this library and pick out a book, say one of my favorite books, I would have a hard time finding it. However, if I went to this library and wanted to find that book, it would be presumably a lot easier. The books are much more organized, there's some cataloging going on, we would be able to find it. So this would be a usable book collection. And in our analogy, this would be a usable genome. Just like when I want to find that book I want to read, I can find it and easily here. Your genome, when it wants to turn on or off a gene, it has to be able to efficiently find that gene and efficiently turn it on. Another thing I like about this picture here that I randomly found on the internet is that indeed the books are nicely organized, but on this work table some of the books are taken out so they can be read or worked on or written about. Somewhere or another those books are being used, just like our genome. Most of the time our genome is completely packaged, but a small percentage of that genome at certain times has to be taken out and laid out on the table and worked with. So I think this is a pretty nice analogy. Now as we move forward, I want to compare just real quickly the difference between chromatin and chromosomes. They often get used interchangeably, but they are different. But they're similar enough that I can see how they get used interchangeably. If we were to draw the cell cycle, where we have G1, G2 over here, and S. And just to remind you, I'm not going to ask this specifically on an exam or anything like that, but it's a good reminder. During this part, G1, S, and G2, that's interphase. During interphase, the cell gets prepared to copy its DNA during S phase, and then it grows and turns on certain genes so that it can enter into M phase, mitosis or meiosis. During interphase, G1, S, G2, the DNA is packaged up into chromatin. And we'll come back to that in a little bit. During M phase, it's packaged up into chromosomes. So what's the difference? Let me list a few. For chromatin, we'll list over here, it will appear as long and thin fibers, whereas chromosomes are very compacted structures. This is what you usually see when you see a picture of DNA, or when they say it's chromatin or chromosomes, they almost always show you the compacted ones, because these long and thin fibers are very difficult to see. Electron microscopes can pick these up, but other than that, they would be hard to see under most microscopes. These long and thin fibers of the chromatin, they are often packaged with proteins called histones. And in a little bit, we'll talk about the different kinds of chromatin that are packaged or unpackaged with histones. Here they're packaged, the histones are still associated with them, plus more proteins. They're highly folded and condensed. They are compacted by about 10,000 times. In fact, to think about how well these are packaged, recognize that in a human cell, there are over 2 meters of DNA. The only way you can get those 2 meters of DNA packaged into a nucleus is by highly and in a very organized manner, folding it and condensing it. In general, we think of chromatin as being metabolically active. And we think of chromosomes as being metabolically inactive. Chromosomes are found during M phase, as we said, and that's the time when we're segregating chromosomes. Very little DNA is being transcribed at that point. So if we think about the central dogma, where we go from DNA and it is transcribed into RNA and then translated into proteins like so. When we say metabolically active, what we're really saying here is that at this stage of transcription, the DNA is able to be transcribed into RNA. Since they are, in essence, the same piece of DNA, we should also point out, just so we point out a similarity, is that they both carry the genetic information of that cell. And same over here. They both do that. In general, we're going to spend most of our time this semester talking about chromatin. We'll come to chromosomes every now and then, but chromatin, this is where the genes are actively turned on or actively suppressed. And so this is really where the work is done. And so we're going to spend most of our time talking about chromatin. So what I'd like to do now is to spend a little bit more time talking about the kinds of chromatin. Okay, I'd like to spend some time now introducing to you the terms heterochromatin and euchromatin. These are both two categories of chromatin, and chromatin isn't either heterochromatin or euchromatin. A long stretch of chromatin, a whole piece of chromatin, at some regions could be heterochromatin and at other regions euchromatin. Heterochromatin we can think of as packaged with histones, and this packaging with the histones blocks transcription. Genes in heterochromatin would be off, turned off. Euchromatin is not packaged with histones. There can be some association of histones near them, but they're not as tightly compacted. Transcription is active. Genes within the euchromatin would be on, or could be on. There might be other regulation that is happening that could turn them off, but they would have the capability of being turned on. Let's say you have a piece of DNA like this. Over here, I'm trying to show you what the heterochromatin would look like. It would have these pieces of histone molecules on it. If there happened to be a gene, say right here, because of it being so packaged, enzymes that would be needed to enter in here and turn this gene on would be blocked. So enzymes do not have access to the gene. Whereas if you had a gene over here in the part that's euchromatin, and our histones have fallen off, like so, enzymes that would like to have access to this gene now have access to the gene, and they could turn this gene on now. We'll talk more about this as we move forward, but a couple things I want to point out to you real quick is that these histones with the DNA associated equals a nucleosome. Nucleosomes contain 8 histone proteins and DNA. The types of histones found in the nucleosome, we have 2 H2As, 2 H2Bs, 2 H3s, and 2 H4 histones. 8 histone proteins in the DNA make up the nucleosome. The nucleosome keeps that gene packaged so it cannot be turned on. I'm going to erase this and I'm going to show you a picture of what the heterochromatin and the euchromatin look like inside of a cell. So in this picture here, what we see, it tells us where the nuclear envelope is, just so we're familiar with where the nucleus is, but it points out heterochromatin and euchromatin. And this dark area here and here, all throughout the nucleus here, that is the heterochromatin. It's very tightly compacted, and so it doesn't take up as much space. But it doesn't mean there's less DNA here. There's probably more DNA in these regions, these darker regions. It's just that it's more tightly compacted. The euchromatin here It is, again, the usable piece of the DNA, the part of the DNA or part of the chromatin that is not packaged. And so it's almost like a ball of yarn that has just exploded, and all the DNA of this euchromatin has been exposed and has access to the transcription machinery to turn on or off genes. We'll talk more about this, but some things that we'll learn is that even at this point, it's not just unraveled in a random mess, it's very organized where it's attached to the nuclear envelope. There are certain regions that have certain kinds of activities. But the way this is unpackaged is certainly not a random event. Okay, quick editorial note here. I originally was going to throw the next topic on this podcast, that is the history of epigenetics, but I'm going to move that to a separate standalone podcast. If I included on this one, it would have made this podcast over 30 minutes long, and I think that's too long. And the two topics don't really coincide to justify them being on the same. At the end of the podcast for the history of epigenetics, I'll include a summary for both of these first two podcasts. So link to the next podcast to continue on to the next objective for this opening topic.